I'd like to introduce our next group of speakers for our keynote. First, we have Mary Millard. She's a patient advocate and sepsis survivor. We have Thomas Lowery, Chief Scientific Officer of T2 Biosystems, and Neil Clancy, who's an Associate Professor of Medicine and Director of the Mycology Program at University of Pittsburgh. They'll be speaking today about antimicrobial therapy for life-threatening infections. Speed is life, and the devastation of a delayed diagnosis. So please come up. Thank you. Mary, you up first?
Good morning. My name is Mary Millard, and I'm a two-time sepsis survivor that lives with a chronic hospital infection. It is Pseudomonas aeruginosa, and I've lived with this for 4 years now. We all know, your pharmacists, your physicians, you're knowledgeable. It's a gram-negative pathogen that's very common in CF patients, but not very common for surgical site infections. I was told that I was one of the only survivors of a graft and valve infection that the IDSA knew of. A little bit about how I got this. I was perfectly healthy, had never had a prescription in my life, was very active. I had some palpitations and went to an emergency room, and they proceeded to tell me that I had a 6.3 centimeter ascending aortic aneurysm due to a large aortic root that was probably thin, and my valve had collapsed partially. I was admitted for surgery, and a day before the surgery, the backwash from the collapsed valve caused cardiac arrest. And that cardiac arrest led to actually coding, and after 3 times with the paddles, it did not work, so I was put on ECMO, which some of you may be familiar with what ECMO is. It's actually machinery that oxygenates your bloods for you and circulates it. I was put on an external pacemaker. 3 weeks later, I received the open heart surgery. I had recovered from the ECMO. And about 4 days after that, I was put into step down to go home. What they did not know during the ECMO procedure, I had been infected. So they graft and valve they had put in received the biofilm that the Pseudomonas creates. My husband came in to talk to me one morning. He found me with my head hanging down, talking in a confused manner, and I had a fever. And this is where the first delay came in. Stroke is very common. After open heart surgery, they called a stroke code. This delayed my sepsis treatment by 8 hours as neuro did a workup on me. And it was a neurologist that finally said this could be sepsis. And I was put back into the ICU. At that time, I was given vancomycin. Does not treat Pseudomonas. I was given a culture, which took 48 hours to come back. At that time, my organs were really beginning to shut down. They had called acute septic shock. And I was finally given the right therapy 2 days later. This resulted in the second surgery where they opened me up again. They tried to debride around the graft. They tried to clean out the chest area and tried to clean it up. This caused an extra 6 weeks in the hospital. I was not awake for maybe 3 days of all this. I learned all of this from records that I read, my husband, doctors, PAs, what happened to me. 2 months in bed and septic shock that seriously weakens your system. I had to learn to walk again. I had to learn to use my hands again, and to this day, I have a numb left foot and tingling fingers and fatigue from the sepsis. And this also resulted in a month of home infusions after I came home from the hospital. I am right now on 1,500 milligrams of Cipro a day. This has been for about 3.5 years. I became bacteremic again 6 months after the operation, where the Cipro, I was only on 1,000 milligrams, and the Pseudomonas overrode that. So another week in the hospital and put on this 1,500 milligrams a day. As we know, it's a fluoroquinolone. It has a lot of problems. I withstood it for probably about 2.5 years very well, which really surprised all the doctors. My kidneys even held up. But again, I started to get tendinitis. I started to get braces on my feet. It was causing other infections and other hospitalizations. And here we talk about what it can do, the cost. Now, I was very fortunate. After this happened, I was put in a research study. My costs get written off because of this. This is cost to the hospital right here. This is what they have to absorb, and I'm just one patient. And we all know that it's billions of dollars a year that they lose taking care of these people. In the past 3.5 years, I've endured 98 x-rays, 30 CT scans. I've had intestinal infections. I've had respiratory infections. I get monthly blood tests and cultures. The problem with this is every time I get a culture, I'm a hard sick. It's painful. It takes a long time to get. But again, I sit on pins and needles, waiting for this to come back, waiting to see if it's back or not. My out-of-pocket expenses are pretty substantial because it requires some payment for medication. My clinic is an hour away. My hospital is an hour away. There's fuel costs. I see 4 and 5 doctors a month. I also get hospitalized at any chance. I'm constantly told to go to the emergency room when something comes up because they don't know what's happening right now. And my chart to date is 324,000 pages. And imagine if you're a new doctor and you're having to look through that. That's crazy. I cannot do anything that I used to be able to do. I can't play sports. I cannot run. I have fatigue. I cannot do anything that requires a lot of effort, heavy lifting. I love to garden. I love to do landscaping. Can't do that anymore. My life has become hospital visits, doctors, and just living with this that is in my mind every day of my life. What is going to happen today? Because I can be hospitalized in a moment's notice for something that has come up. And this really interrupts my day. Initially, my husband had quit his job because when I was in the hospital, he was told every day, this may be her last. And what would your spouse do in that case? They'd want to be with you. So it has been devastating, and he has become my primary caretaker. He worries about me so much now that I can't go anywhere without him wanting 20 texts exactly what I'm doing and how I'm feeling. So it's pretty devastating. And it's also, the septic shock can also cause a lot of health issues. It's caused heart failure because of the oxidative stress that the heart goes through. So I've had a pacemaker insertion right now. That little thing right there costs $89,000. And I've also gotten skin cancers due to the photosensitivity from the Cipro, both BCCs and SCCs. And I want to thank you for your time today. This is very important. I think the patient voice is important. People don't understand that this can happen to anybody. People don't understand that this changes your life forever if you're living with it. Most people die, but there are survivors out there. And it's very difficult. And the work that you people do is very important. And I want you to continue it. And I want this Congress to grow. And I want to make sure that everybody hears that AMR is such a devastating thing that we have to address this up front and now. Thank you.
Thank you, Mary. I'm sure I don't speak alone, that's very motivational for all of us that are involved in combating AMR, and I want to thank Larry and Carrie for their calls to action as well. I just want to take a few moments and talk about what we're doing at T2 Biosystems to help address the issue of AMR and what we hope our technology can help do in getting patients on appropriate therapies earlier. So our technology is intended to complement the standard of care to help identify patients who are infected with pathogens and have bloodstream infections far earlier than what is available today. The panels that we have available, we have 2 panels that are available for use in clinical care here in the United States, the T2 bacteria and the T2 Candida panels. The T2 bacteria panel is designed to cover the most prevalent pathogens that are known as the escape pathogens that are most commonly not on appropriate antibiotic therapy. And the T2 Candida panel is meant to address the problem of candidemia. We also have a panel called the Candida auris panel that's looking at the multi-drug resistant form of Candida that's changing hospitals' epidemiology internationally, and also is being monitored very closely by the CDC. They validated it for patient monitoring with a skin test. And then just a couple weeks ago at ID Week, we announced our first resistance panel, also direct from blood, for detecting pathogen-resistant genes, the carbapenemase resistance genes, as well as a genus-level channel for Klebsiella and Enterobacter. And then we have another panel that's in development that is a gram-positive resistance and ESBL panels. And those 2 panels are really grateful for the opportunity to work with Carbex. We're powered by Carbex to develop these resistance panels and help speed up that time to knowing that your patient has a complicated infection, a bloodstream infection, and a resistant infection directly from the blood sample. So if there's one thing I hope that you take away from my talk is the T2 Biosystems, our focus is to provide results directly from the blood sample. So today in the field of identification of pathogens and also identification of resistance, we're all waiting on blood culture. Blood culture is a technology that's been around for decades and decades and has some serious limitations in terms of infections that it's missing, as well as interference from antimicrobial therapy. And so our technology takes the blood tube directly from the patient, 4 ml EDTA vacutainer, and that can be loaded on the instrument, processed in an automated fashion. Within 3 to 5 hours, you have a result. And it's the only technology that's been cleared for use here in the United States, and the outcomes data and the data is now starting to build in terms of how this can really be a paradigm shift for hospitals, for clinicians, and for antimicrobial stewardship committees to make appropriate therapy decisions far earlier and have greater impact on patients. The instrument itself is fully automated. It's a random access analyzer, can take up to 7 tests at a time. It's configured for a laboratory to be able to process those samples as they come in. The EDTA vacutainer is simply snapped onto the cartridge, put on the instrument, and then loaded, and then you walk away and the result then is available in the LIS or printed out before the instrument, the cartridge is taken off. We now have about over 160 hospitals worldwide that have access to our instruments in their laboratories and are using them, and then the data is building for this type of testing and how it can impact patients. So one thing I want to just comment on, I'm not going to go into all the details before I turn the time over to Dr. Clancy, but there are building data now showing clinical sensitivity and specificities that are very actionable. Clinical sensitivities for all of our different pathogens are in the range of 90%. Specificities are in the range of 98%. That really provides the confidence to be able to make therapy decisions much earlier in that time of progression of disease for the patient and help get those patients on appropriate therapy and withhold therapies that are inappropriate far earlier than is currently possible when we're waiting for blood culture results. The last data that I want to show is just some data from our pivotal study. A lot of times people say, particularly when they go and talk to the laboratory, wait, but those culture results do come back fast. So this here is data from, we had 11 centers participate in our pivotal study for the T2 bacteria panel. These are the 5 different pathogens here shown in blue, are the time to result using our platform, just in a time frame of about 3 to 5 hours. And then shown in orange here are the actual time to species identification. This is based on blood culture and then MALDI-TOF. So you can see a substantial difference in that 5-hour turnaround time versus 3-day turnaround time for that species identification. And so being able to use a technology like this to direct your therapy early on really can help eliminate the guesswork in those critical hours that are important to help truly impact patient outcomes. So with that, I'd like to turn the time over to Dr. Clancy to speak more about clinical use of our tests.
Well, great. Thank you, Tom, and thank you to the organizers for the opportunity to participate. And thank you to Mary for sharing her story and for her advocacy. And this is a big topic of discussion yesterday, the need to get real sort of life patient stories out there. I think those of us who take care of the patients even lose sight of sort of the individual and the personal toil that these take. And I commend Mary. She's all over YouTube as well and fighting a good fight. So I think her case teaches a number of things. There'll be a handful, but I'll just talk about real quickly. And the first lesson is that for all the talk about surviving sepsis, really that's just an early step on what is a long road. And if we look, at the data from the U.S., there's 20 million patients with sepsis. 70% of them will survive to hospital discharge, but as Mary has highlighted, half of those patients have long-term complications and sequelae. So if you look at survivors, 33% of survivors are going to die within 12 months, and then of the remaining survivors, 16% will have persistent medical problems, and Mary ran through just about all of these that are listed here in her own personal case. I think the second lesson is that sepsis incurs massive costs. There are systemic costs. So, for example, it's been shown that it's, in fact, the most expensive condition that we treat in U.S. hospitals, and estimated economic impact of $23 billion to the healthcare system. But in a sense, numbers like this are almost so big that they're hard to even know what they mean. And I think when you look at sort of the individual patient level and what it means to families and loved ones, as Mary has detailed, that I think it really hits home in a more powerful way. So in 2018 US dollars, the median cost per patient per month following survival of sepsis on the outpatient basis is about $1,700. So this obviously quite adds up, and in a number of cases significantly higher than this. The third lesson, and Tom touched upon this, is the importance of time to accurate diagnosis and initiation of appropriate antimicrobial therapy. And this is absolutely crucial. There's a large body of data showing that even an hour's worth of delay starts the clock and results in worsened outcomes. Kumar is probably the most cited study. From the time of the onset of hypertension, low blood pressure, at the start of sepsis, each hour delay in therapy increases mortality on the order of about 7 to 8%. And if we look at Mary's case, let's say at 8 hours after they had done their stroke workup, they had actually started effective therapy for sepsis. Even then, the survival rate shown in the black bars here is about 33%. So it's quite remarkable that she's even here with us. But of course, in her case, the therapy was delayed 48 hours for the cultures. And it's truly remarkable, you see, because survival in that situation is essentially nil. And then the final point that it brings home, and again, Tom mentioned this, is cultures are wholly unsatisfactory, but in fact are our diagnostic gold standard. And it's not only the time to pathogen identification and susceptibility, but it's the inherent insensitivity of them. So if you look at various forms of suspected sepsis, suspected bacteremia, sensitivity is on the order of about 10 to 50%. If you're talking candidiasis, we know from autopsy data, it's probably about 40%. So we're missing over half the cases as well as having to wait a long time for results to come back. And it's this, the recognition that time to therapy is crucial and our existing diagnostics are not up to snuff, that's fueling the widespread empiric broad-spectrum antibiotic use that's contributing to resistance. So, that's the reason so many of you are engaged in rapid diagnostics, and a lot of us are engaged with integrating rapid diagnostics into patient care and stewardship. So here's the T2 candidate panel. FDA approval was based on the DIRECT-1 study, which was published 3 years ago now. Since then, there's been the DIRECT-2 study, and the T2 candidate panel, the 5 species that it covers account for 95% or more of invasive candidiasis in U.S. hospitals. Tom mentioned overall sensitivity and specificity of 90 and 99% respectively. And we know it identifies many of the patients who are blood culture negative that we're missing with the existing diagnostics. And it's particularly useful in those patients who are receiving antifungal therapy at the time of testing. So antifungals will blunt your blood culture, but this technology is able to detect at least some of those patients who are already on antifungals. And this is absolutely crucial because in sepsis, you want to get the antibiotics on, and not uncommonly, diagnostic tests, despite best intentions, get drawn later. So the inability to detect blood culture is a real limitation. And if you actually then do the math and you figure out how would this test perform in the patient populations who are at risk for candidiasis, what we're really interested in are the positive and negative predictive values. How likely is it if I have a positive test the patient really has the disease, and how likely does the negative test mean that the patient does not have the disease? And if you do the math, you get positive predictive values in our target populations of about 50 to 80%, and exceptional negative predictive values of greater than 99%. So negative tests, almost without exception, rule out disease due to these pathogens. So the escape pathogens, these are our major concerns in terms of antimicrobial resistance in hospitals like ours, and Tom ran through this: E. coli, Enterococcus faecium, K. pneumoniae, Pseudomonas aeruginosa, and Staph aureus. And just to point out, if a test like this had been available in Mary's case, 100% sensitivity in the pivotal trial data for Pseudomonas, for example. Again, from the pivotal trial data, if you look at specificity, if we take those proven cases where the blood culture was positive for one of the pathogens detected by the panel, the specificity was 90%. So basically identical to the T2 Candida data on a per assay specificity, so looking at the specific pathogens, specificity of 96% for E. coli, and then greater than or equal to 98% for the other pathogens. If you go beyond that, though, and also look at proven or probable cases, these are the patients in whom blood cultures are negative, but there's a clinical indication of infection, and cultures oftentimes at other sites are positive for the pathogen. The specificity actually increases to 94 to 96%. And once again, that's for T2 Candida. T2 bacteria identified a large number of these probable possible cases that are blood culture negative, and the real benefit was in patients who had already been started on antibiotics where T2 is positive, but the blood culture is negative. So that's all fine and good, sensitivity, specificity, clinical trial data. How do you implement this on the ground in clinical practice? And this is where I think the clinical community really needs to get a bit more on the ball and think through these, because they're complicated algorithms, but they're workable. So our pilot program for T2 Candida was directed towards sepsis in our medical intensive care unit. This is a population at UPMC, our hospital, where 3 to 5% historically would be diagnosed with candidiasis. So what we put together, and Hong Lin has really led this charge at Pittsburgh. She runs our stewardship program, and we've established a number of diagnostic stewardship teams in specific units directed to specific clinical scenarios. And what you can do is you can use either the positive or the negative predictive value then to come up with positive early empiric treatment based on a positive result, or if you've already started patients, using the negative predictive value to discontinue therapy that you've initiated. And then you can run through sort of the algorithm about administering drugs and folding your blood culture results with that. So here's the pilot data for the first 125 patients in the MICU with sepsis. Using both the T2 Candida and blood culture, we identified 15 patients with Candida sepsis. If we had only used the blood culture, we would have identified 7. So that's pretty well spot on. If we say the sensitivity of blood culture is 50%, that's exactly what we would have expected. And what we've shown is that overall antifungal usage since the initiation of the program has been reduced 47%. So then T2 bacteria is a little earlier in the game. FDA approval has just come down the past couple of months. So how do you develop a strategy for this? And this is what we're working through right now. So if we look at the 5 pathogens on the panel, at UPMC, these will account for 2/3 of gram-positive bloodstream infections. If we exclude contaminants like coag-negative staph, the bacillus, and what have you. If we look at our gram-negative bloodstream infections at UPMC, what we find actually is that most of these cases are diagnosed immediately upon presentation or within the first 24 hours of hospitalization. So these are patients in the emergency room, admitted directly to your ICU, or within 24 hours are transferred from medicine surgery floor to the ICU because of sepsis. And if you look at this early sepsis, 70% of sepsis due to gram-negatives within the first 24 hours are covered by the gram-negatives on the T2 bacteria panel. So our focus then is ICU, ED within the first 24 hours of hospital admission. That's where we're going to go after. So then how might you develop algorithms based on incorporating these data, given the fact you're only detecting 5 pathogens? Well, in fact, if you look at the implications for stewardship, these are the 5 pathogens that drive a lot of our broad-spectrum antibiotic use and excess treatment use. So you might envision at the University of Pittsburgh, we have a good idea where are carbapenem-resistant Enterobacteriaceae, which is largely at our place going to be KBC-producing Klebsiella. We have a good idea where our high-risk populations are for Esbl-producing E. coli. So if you target testing to those populations and you detect a pathogen, what you can do is, if you get a positive test, you can direct your big gun, broad-spectrum, new beta-lactamase inhibitor agents it's negative, then you can go back to a more conventional treatment. And you can do similar things in the gram-positive space. And the other thing, if we get a mixed result where the T2 is positive and the blood culture is negative, at least initially, we believe the T2 result, and we guide treatment based on that. So in conclusion, I think these T2-MR panels are important advances for the field. Both T2 Candida and bacteria have excellent performance in clinical trials. Now we've got to figure out how to put them into practice. They have shorter turnaround times and improved sensitivity compared to blood culture. But I think the key thing here is you can build intervention strategies that exploit both positive and negative predictive values to either start or discontinue therapy. I think it's important to validate these strategies. They look good on paper, but what do they actually do in the field? And we're very excited, as Tom alluded to, about the incorporation of resistance panels, since we know where we're seeing our E. coli and our resistant Pseudomonas and Klebsiella. If we had rapid detection of resistance determinants for those pathogens, I think we could streamline stewardship even further. So with that, just to acknowledge our team, and particularly Paul, who runs the show at Ryan Shields on the pharmacy end, has coordinated the unit pharmacists in running the diagnostic stewardship program in the respective units. And with that, I don't know if we have time for questions. We don't. So the idea was to have a little 5-minute thing, but I think we'll all be around for the rest of the meeting and be happy to chat. Thank you.